Seth Crosby. I'm the Director of Scientific Collaborations here at MGI, and this is the first in a series of what we hope will be monthly lectures concerning the activities at the McDonald Genome Institute here at Washington University. The Institute is comprised of a multitude of scientists, labs, and facilities involved in a broad spectrum of research. Today we'll be hearing from our genomics facility, GTAC, at MGI. This will be presented by its director, Richard Head. Rich has been immersed in medical research for over 25 years in both academic and industrial settings. Most of his work has been in the development and use of high-throughput genomic technologies, such as microarrays, sequencing, and high-throughput PCR. He and his teams have successfully brought these technologies to bear on the discovery of novel drug targets and modes of action, biomarkers for patient stratification, development of preclinical models, and ascertaining the causes for drug failures. While at WashU, in addition to leading our facility, he has invented 2 published computational suites, one which he will discuss today, the other significant advancement in the use of 16S sequencing in the characterization of microbiome communities. If you have any questions, feel free just to fire them into the chat box, or at the end of the talk, I'll be unmuting the audience and they can raise their hand. If you look at the bottom of your screen, you can see the little hand raising icon if you'd rather speak your question rather than type it. We're going to be recording this seminar for posterity, and I'll upload it somewhere on YouTube to make it available for people who are not here today. And with that, I'm going to mute myself and let Rich take over.
Thanks, Seth. So yeah, today I'm going to talk. We used to do these before sort of pre-pandemic and pre-GTAC MGI merger. We used to do these annually, and now with a number of new technology centers evolving, as Seth mentioned, within the MGI and working synergistically in some cases, we thought it would be a good idea to start these back up, to start basically just making sort of official announcements about new technologies as they're becoming available, when they're becoming available, who you need to talk to get access to them, and so forth. So today, I'm not going to talk too much about the, the stuff that GTAC does on a daily basis, because I think a lot of folks know that. And if you don't, we have a website where you can get a lot of our standard services and stuff that we do both in the lab technology and bioinformatics space. But today I'm going to focus a little bit more on some of the new things that we have going on. Some of these are released now, some will be released over the next month or two. Virtually everything that I talk about today will be available by this summer. So we thought it'd be a good time to start putting that out there so people can start thinking about these new technologies if they want to start utilizing those in their research. And it's not a bad time to come talk to us before you really get started, even with some of these, just to kind of know what the sample requirements are and so forth. So I'm going to give a very brief update on our integration update of all of the legacy GTAC and MGI systems. I'm going to spend pretty much the lion's share of the time on discussing new lab technologies. There's a couple in particular that we're pretty excited about finally being able to make available. A couple of new things on the bioinformatics fronts in terms of some new releases, as well as a new collaboration portal that we announced about 2 weeks ago. In terms of some of the infrastructure improvements that we're making, these are specifically designed to improve interactions with customers and collaborators. So I want to point these out in terms of what's available now versus what's coming over the next month or two, how we're trying to streamline and improve our interactions and our ability to track samples and projects. And then also just kind of a brief primer to the upcoming technology seminars that Seth mentioned. So in terms of our progress towards the full integration that started about 3 years back now, we have, in terms of the plan that we had put forward when we did the integration, we wanted to streamline the operations of GTAC and the McDonald Genome Institute, particularly with regards to the genomic technologies. And we want to do this for both the wet and the dry. And when I say streamline, I mean try to basically build improved workflows that allow us to get data through faster, more cost effectively, et cetera. So that was really what drove our goals, improving basically cutting edge technology access, improving our turnaround times, decreasing costs for services, and also hopefully generating tools that improve the return on investment for the data that's being generated. The pandemic delayed our progress a bit, and this was due in no small part to the fact that, as many of you may know, we developed a saliva test that was used here at the institution for the last 2 years, in a number of different ways, as well as the state of Missouri and the Radix grant teams for a number of the special needs school districts here in Missouri. So, you know, we were very much involved in that. We've processed, I think, well over 200,000 samples with that saliva test that we co-developed with Fluidigm. But as you can imagine, the fact that we had to shift personnel and equipment for that really did delay a lot of what we were trying to do. And then supply chain issues have been a problem. I'm sure everybody's dealt with that. So, and our IT infrastructure migration has been a very substantial undertaking. We are now moving into the new research computing environment with everyone else. So we're slowly moving off of the legacy MGI compute infrastructure onto the research computing environment that will hopefully help streamline our ability to not only process data more quickly for people, but also return it more effectively within the community. So just a brief update on that. And so now let's get into what probably people are most interested in, and that's some of the new technologies. So as probably many of you know, we've been running single cell in a production format here using the 10X Chromium system now for quite some time. This has been going on in Jen Ponce's lab. And this system basically enables single cell gene expression, as well as some immune profiling and TCR with respect to TCR and BCR. It also allows a multi-ohm ATAC plus gene expression, as well as an ATAC only. So this is all stuff that I think folks are mostly familiar with, and most of this has been up and running for a while, working fairly well. The Cell Ranger software workflow that comes with this is fairly standard, fairly usable. Plus, we also have Seurat and Monocle and other workflows built into our bioinformatics pipelines. Now, what's coming new here is that we have purchased the high throughput Chromium X or Chromium 10, depending on how you read it, system, which allows us to target 20,000 cells instead of the 10,000 without multiplexing. So it's a much higher throughput system. It can process up to 16 samples at a time instead of 8. And full runs of 16 will likely have some price discounts associated with them as compared to partial or current runs. So we're still working on that, getting that pricing established. But once the system goes live, we will have that established. So this may actually help enable both larger experiments in terms of the numbers of cells that you want to process, as well as the throughput, and should hopefully have some cost benefits as well. We have our training scheduled for mid to end of this month with the system that is in-house now. And our target release date for this new high throughput system is May 1st. So this should be available after that point. In addition to that, we've also been working with Tristan Lee's lab to bring online SmartSeq. SmartSeq is a very complementary single cell protocol. It will be replacing GTAC's legacy FACSeq technology. For those who have used that or are familiar with it, if you're not familiar with it, this is a plate-based single cell technology that allows you to basically use FACS sorting to put a single cell into a well in a 96 well format. The old FACSeq was 96 well, was as good as we could do. SmartSeq is, let's say, a more robust and kitted version of the technology than what we ran here. And it is, like I say, it's complementary because it works well when you have a limited or specific cell population that you want to profile deeply. So with droplet technologies like 10X, you typically will get 5, maybe 6,000 transcripts back, no matter how deep you sequence the samples per cell. With the SmartSeq and FactSeq technologies, because of the way that the library generation is done, it's a little bit different. You are actually able to, if you sequence deeper, you can detect more transcripts. So it's a more sensitive platform for doing a very thorough characterization. And if you're working with a very limited number of cells that you are able to fact sort, you can isolate those cells individually and use this protocol. So like I say, it's been a very complementary technology to the droplet technologies, but the SmartSeq version of it is higher throughput and lower cost. It also offers full gene body coverage. It's not just 3' or 5' sections. So even our FactSeq was very 3' bias, and the SmartSeq technology will allow full gene body coverage to be seen. So it definitely has some advantages over the droplet technologies. Like I said, in a complementary way, not a competitive way. And our target release for this is sometime probably in the June timeframe. Now, there's 2 parts to this. The 96 well format we will probably have ready to go before June. The 384 well format, we're probably going to use the SmartSeq 3 protocol for that, as opposed to the SmartSeq 2. And so that probably will be more of the June, possibly a little bit later timeframe, but basically in that ballpark is what we're looking for there. Now, the other new technology that we're bringing on, and this is something we've not done before in GTAC, is spatial transcriptomics. So we have purchased, or basically we've invested in the 10x Visium platform. It's a slide-based spatial expression profiling system, which uses much of the 10x technology that we currently have. And you can do with this, as opposed to other, some of the technologies for spatial transcriptomics are targeted only. Visium is what's referred to as whole transcriptome. It is 3' bias, much as the 10x technologies are, but you can detect, it's not limited to a fixed target space. And it will work with fresh frozen and FFPE. I've got a little star next to the FFPE to remind me to tell you there is a QC step that we run when working with FFPE, because the DV200, the percent of nucleotide sequences above 200 bases in length, needs to be above a certain threshold in order for it to work. But, you know, basically any reasonable FFPE that's probably not, you know, greater than 6, 7, 8, 10 years old has a pretty good shot of working here. There are some protein code detection capabilities that you can run with this, and there are human and mouse kits. The current resolution of the system is just under 5,000 spots per capture area on the slide, and there are 8 capture areas on the slide. And the spot diameter is about 55 microns. So it's a little bit big. You're not going to get individual cell level with the current resolution. However, we've been told that the HD, the high definition version of this, is anticipated to be coming out sometime around this summer. And that goes down to 4 microns. So that will get you individual cell level resolution in a lot of cases. So this is a platform that we're going out of the gate with. We are looking at other complementary technologies such as Visgen, Merfish, and we've looked at some others as well within our Tech D group here, Bob Fulton's group. And so we are looking at potentially something as a, much like SmartSeq is a complementary technology to a, let's call it a higher throughput screening approach like 10x. We're looking for something here that might be higher resolution, but maybe in a targeted way. Because again, once you get to the higher resolution platforms here, you tend to you tend to have to work in a targeted fashion, not whole transcriptome. So again, we will likely look for a second technology to bring online in space as a complementary platform. In terms of software, there is software available in which you can, and there's multiple features to it. That's not a typo. They actually did call the software for Spatial Space Ranger, as you might expect, given the old version was Cell Ranger. So here they went with the obvious Space Ranger. I guess Disney didn't call them out for that. So the software is freely available, as I understand it. It has data and image processing. The clustering and gene analysis is what you might be very familiar with, at least in terms of the types of outputs that you see in regard to, you know, stuff similar to the TS&E type plots. You can actually see some of that down here in the lower left. So, I mean, visually, these things will look somewhat similar to what you're used to, but you have the ability, as I understand it, I haven't used the software yet myself, you have the ability to overlay the data coming out from the transcriptomic analysis with the your captured slide from the microscope. So you kind of have this flow again to be able to see the clusters, see where where those clusters exist, and then break it down to the individual genes. And I believe there is an overlay function within the tools so that you can use your H&E image to overlay with that. So this again is all software that is currently available with this Visium platform. So we are currently in testing. We're doing both mouse and human with our testing. And we, like I said, the FFPE is doable in this space. In fact, I think one of our tests are actually, one of our initial tests is with FFPE. And we've already done some of the DV200 estimates on that we'll be doing in the first sort of test or pilot experiment. There are two options for creating the slides. GTAC is partnering with the DDRCC in order to be able to provide this as sort of a full service slide generation and processing. So, you know, experts basically who have histology technicians that do this stuff every day know how to do this, have the specialized equipment that you need to make these slides appropriately. So that will be offered as a full service, like I said, through our partnership with DDRCC. You can also do the slide prep in your lab, but I put the little star there because it does require specialized equipment and expertise for making these. So that's just something to be aware of. I mean, certainly we have labs here at WashU that are very heavy users in the single cell space that have their own Chromium systems for doing single cell and then send the libraries over to us. And so if you have the kind of equipment that's necessary for making these slides, by all means, we'd be happy to work with you in that format. Our approximate availability or estimated release is June for this system. And because the HD version is expected sometime this summer and will work basically with all of the same infrastructure, it's basically just a higher definition version of the chip. As I understand it, I don't think there's too many changes to the protocol itself. Once that becomes available, that will probably go online in production fairly quickly thereafter within our single cell and spatial labs. Okay, so the next kind of shifting gears a little bit from spatial and single cell, the SomaScan platform, which we've been running here at GTAC for over 5 years now. I think we started 5.5, 6 years ago with it. Some may know that at one point Soma was going to exit the service business, but they changed their mind. And not only have they changed their mind, they've changed it in a very big way. So the system we ran here for 5 years and generated, I don't know, somewhere probably north of 50,000 arrays, or somewhere in the ballpark of 25 to 50,000 arrays in that time, which was about the capacity of our lab. And they have come out now with a 7,000 analyte version of the chip. So 1,300 proteins is what we used to get, or 1,300 protein analytes. Some of the there were different forms because this is a conformationally dependent assay in terms of how the DNA aptamers bind to the proteins that are used in the signal detection process. The system will sometimes have 2 or 3 or 4 forms of a given protein, especially proteins that have extracellular modifications made to them. For example, fibronectin has a number of different external cleavage sites, and that creates conformational changes. And they actually had several different fibronectin forms that they recognized on the platform. But this new version, 7,000 analytes will be available. Currently, we have the kits in-house for serum and plasma, and that system is up and running. It went live in basically December. We're just now officially announcing it because we wanted to get through some backlogs of samples that people had held from the 1300 analyte platform to the 7K. And so we've been processing those, and I think we're mostly through these now. So we are officially announcing that SomaScan 7K platform is open for business at GTAC, and we are able to handle samples on an even larger scale than we were in the past due to the robotics and automation that are now part of this 7K platform. So the system, and I'm going to say 10 log range here in quotes. The way they have built the 10 log range sensitivity within the system is that they know the approximate ranges in picograms per mil of concentrations of proteins that you typically see. And they've built the platform to be able to measure those proteins within those ranges. And that covers about a little over 10 log units. So the interesting thing is the reason that there's a limitation at all is because this is done on a microarray scanner. This is effectively the DNA aptamers that they use to bind and pull down the proteins that they eventually pull off those aptamers to bind to the microarrays. It's effectively like a cDNA microarray to measure the signal. They have announced that they have partnered with Illumina, and over the next roughly 1 to 1.5 years, they are going to convert the technology so that you will be able to use the sequencer, the Illumina sequencer, to process these same samples. That technology won't be available supposedly until about late 2023, as I understand it. But given that we are an early access beta lab for this technology, I suspect as long as our relationship continues with SomaScan moving forward, we will probably have early access to the Illumina technology as well. In terms of the utilization and impact at WashU, this platform, as I said, has been used for 5 years, over 5 years. There have been numerous high-profile publications, both internally and externally. There are just a few examples here from Jeff Gordon's lab, Doug Mann's lab, Carlos Grishaga, in Science, Nature, Nature Neuroscience, JACC. These are just a few of the high-profile papers. There are many, many more that have been published both internally and externally over the years. It's been a very productive platform, especially in its ability, especially using fluids like plasma and serum, urine, CSF, in order to detect proteins coming from tissues where if you didn't biopsy that tissue before to do RNA-seq, you wouldn't have been able to see these expression changes easily. And so the sort of the broad screening capabilities of the platform are quite useful in being able to detect things from secreted proteins that leak from tissues into those biofluids. As I mentioned, the system is installed, it's up and running. We can now run, the cost is only marginally higher than the 1300 array platform. Internally, I think it's only roughly $25, $30 more an array, even though it's over 4 times, almost 5 times the content. And we can do up to 85 samples per run. So we're actually working, if for those who've used it in the past, you may remember we required you to be able to do at least 24 samples per run because we didn't have a way to kind of mix and match from different labs. Because we know a lot of labs will not have 85 samples to process at a given time, we're working on a way to be able to handle that and work with labs where we're combining samples from different projects into a single run, because we know that would be very, very limiting for labs, especially when you want to do a test or a pilot and you've only got 10, 12, 15, 20 samples that you want to run in order to do some sort of pilot experiment or small experiment. Currently, we can only do the serum and plasma, but I believe, as I said, the kit that will allow for cell and tissue and or other fluids will be available this summer. At least that's our understanding. We don't know exactly when. They haven't given us a firm date yet, but that's the projection. And as of right now, we are one of only 3 labs in the United States up and running with the 7K platform outside of SomaLogic themselves. So we were one of the 3 early access beta labs for this platform. And as I said, we've been running it since December, and the results so far have looked very, very good. Additional services that are on the near horizon, we are going to be offering some high throughput DNA and RNA extraction protocols. Here back at GTAC, we had gotten down to where we were really only doing a couple of these, and we've had requests, particularly certain types of requests that are somewhat selective to what gets done here. So we work a lot with the TPC and the Hope Center for doing extractions, but and we will continue to do so. But there are certain things like we get requests often for high throughput PAX-gene blood extractions for RNA-seq. Just as an example, there are a few others, and we've already brought a Kingfisher system into our lab, and we will be bringing that online. We anticipate availability for that sometime this summer, possibly early fall. It's probably going to be staggered. We'll probably bring on certain protocols at different times. So it's possible we might even have one of our really highly sought protocols, possibly PAX-gene blood for high throughput, like I said, for RNA-seq, because we get that quite frequently. We may bring that online more quickly if we can. So that's just something to be aware of, that we are going to bring on some extraction services that are, again, complementary to some of our sibling centers across the institution, and particularly for things that we do here. We're, in terms of long read technologies, we are looking to invest in the Oxford Nanopore system. For those who've worked with us, you may know we already have the PacBio Sequel II instruments for long read, but given some of the advantages of the Oxford system, we're looking to bring that online as well, and again, run both platforms for the foreseeable future. Their error rates for Oxford Nanopore have reduced significantly over the last year or two. It's a very cost-effective platform if you are able to work with the error rates they do have. And so we're in, and I think the read lengths can be in some cases significantly longer than what you can get from PacBio. So we're going to be purchasing a Promethean and maybe a Promethean P2 system. We're still determining that. So we're anticipating having this system available along with the PacBio Sequel II system by this fall. Okay, so that's a lot of what we have going in the lab space. In terms of bioinformatics, there are a few things that we have brought online or bringing online very shortly in that space that may be of use or value to a number of folks. So 2 weeks ago, we announced the full launch or formal launch of a bioinformatics and computational biology collaboration and resource portal. Now, we are maintaining the site, but it's intended to be an institutional resource. We are seeking collaborators or contributors, I should say, from across the institution. What this portal does is it provides a mechanism for individuals or laboratories seeking contributors who are experts in the bioinformatic or computational biology space for collaboration. And it provides a way to link laboratories and contributors in a way that doesn't basically, it keeps the research that you post that you want help for is only visible to contributors. So it's not like the research you're seeking help with gets posted out across the whole institution, or certainly not outside of the institution, as the site itself is within WashU only. So for anyone who's listening to this who's outside of WashU, I'm afraid to say this particular thing you can kind of not listen for a minute, because this is only at the moment available within Washington University for investigators here as well as contributors. We may eventually make this available to partner institutions like SLU and Mizzou and some other places that are involved with CITEMAN and ICTS grants. But at the moment, you have to be within the WashU firewall, or you have to have VPN access. And again, as I mentioned, if you have a project that you've done, say, RNA-seq or SomaScan or some other type of omics technology that you've used, and you're seeking help bioinformatic or computational biology help in the form of working on a paper, just doing your research, help on a grant, something along those lines, you can submit the request. That request is then seen by all of the expert contributors who are registered in the system. And if one of them is interested and available, they will contact the investigator's lab and see if there is a collaboration that can occur. And again, there's a basically buttons and toggles for all of the different types of collaboration that you might be seeking, the types of data, et cetera. And there's even additional information about the time frame that you need help on and so forth. So even with all that, it probably takes about 5, maybe 10 minutes tops to fill out the request form and submit it. In addition to that, there is a whole resource directory of educational opportunities offered by many labs here at Washington University, software and tools that are offered by different labs that are available to WashU investigators, pipelines and workflows. Some of these links will go outside of the university to open source type resources, but as you will see when you look at it, there are quite a few internal resources as well that are linked. And this is growing. We have contributors currently coming from individual WashU labs, from the Institute for Informatics, as well as GTAC. So this contributor list is growing. As the contributor list grows, we're also growing the types of bioinformatic computational support that is being offered on the site. We actually had a new contributor join shortly after the last talk we gave on this, who is an expert in the protein structure space. And so that's basically, we're going to add that on as a new type of collaboration request on the site. So as this contributor list grows, so will the opportunities for collaboration on the site. CompBio, some news there. CompBio is the platform we built here at WashU. And even though it was commercialized outside of the institution, it is freely available and in an academic version for all WashU investigators. It's also available to WashU collaborators for projects you may be collaborating with folks outside of the university on academically. It's not, if you're commercial, I'm sorry to say you'd have to go through Perchaya to get access to it. But if you have collaborators, if you're here from WashU, you have collaborators outside of WashU or who are academic or nonprofit, they also can access the system. We have an external facing version of it for that. We have about 200 labs here at WashU that currently have access to the system. And we have now publications that have used and cited CompBio in Nature, Cell, a host of Cell journals, JACC, PNAS, and many, many others. Those are the ones I've listed are just some of the high impact journals and papers where it's been cited now. So it's being accepted, even though it's a new platform and relatively new, 3 years old now. There's plenty of publication out there now citing it. So it's very easy. The information, the type of stuff that we know how to put into the papers gets accepted quite easily now. Expected release for version 2.5. So there's an update coming with some new functionality. We expect that to be released by May. That knowledge base will be created from the 2022 release of PubMed, which has actually over 30 million abstracts and over 3 million full text articles that are ingested into the system. And probably one of the longest awaited features for individuals who've been asking for this or use the tool is an auto annotation system. So I'll mention this in just a second in a bit more detail, but it will be a one button click that will allow you to annotate a current project or even your older projects. We also now have an entity theme content mapping tool. It's just a button that you can click from a bottom tab on the screen. And what this does is it allows you to see what % of your genes or entities, whether genes, metabolites, microbes, whatever, that you put into the analysis. It tells you what % of those have been uniquely mapped by what theme. And so what this does is this allows you to graphically see where most of the primary biology has likely been mapped. So in this particular case, you can see 80% of the genes have been mapped to some type of biological process by theme 9, and 90% have been mapped by theme 20. So basically anything past theme 20, even if it's statistically significant, is very likely secondary, tertiary biology related to these genes. So basically it gives you a really nice focus window to know how far you really need to look out in the statistically significant themes that the tool produces to know where most of the primary biology is being described. We have a number of enhanced interface features that will be available as well in terms of sorting and project management and so forth. And we have a number of additional project level comparative analytics that will be coming online soon as well. So the auto annotation feature, again, we've been getting many requests for this for a long time. It will be a single button system. It will probably be up here with most of the other buttons, or you just say, click a button and say auto annotate. And the thing that makes it really useful is, well, one, it's a lot faster than even having to use the current semi-auto annotation system, but it's better in a lot of cases even than human annotation, because what the system does is it doesn't just look at one theme at a time to come up with the best annotation for it. It looks at that theme and then it looks at its neighboring themes, and it uses the information, the undercurrent of the biology around that theme to identify the most likely description for the biological processes, pathways, representations within that theme. So it basically uses the entire connectivity matrix in understanding what the likely best description for the biology within that theme is. And it will allow for more than a single label, because as those who've used the tool know, sometimes it will bring back two very closely related processes. And sometimes you want to label both processes, and the tool does that in an automated way. It looks for non-redundant annotation, which is something you can't get even in most of the pathway tools that are out there. You'll often get the same pathway back 15 times under slightly different names. This actually will help remove any sort of redundancy in the system, even within the theme. So again, this will be part of the release that will be coming out in May. In addition to that, we often get asked, and this is not necessarily related or only tangentially related to CompBio, we often get asked, what are the best tools, pathway tools to be using for some sort of analysis that somebody wants to do for, their omics data. So if you're using gene expression data, which is, obviously one of the largest types of data used for this, but now protein data are becoming pretty prevalent here as well. It's not an easy question to answer, because most of the analyses that have looked at these tools in the past have not really looked at them from the perspective of biological accuracy. Most of the analyses have looked at p-value distributions and how many things came back, and not necessarily like a fairly rigorous assessment of biological accuracy. So our team has come up with an analysis approach that allows us to take different tools through a very rigorous assessment of basically biological accuracy of the tool. What are you getting back? How much are you missing? And it turns out that the differences between these tools are dramatic in terms of how much you might be missing. So there's literally a half a dozen different assessments that we do. And we're going to publish this obviously at the end of the day, but we will put probably all of these data up on the Bioinformatics Portal site that will just be available for anybody to look at when they're making an assessment of what platform they might want to use. And what we're doing is, for the current assessment, we're looking at GSEA, IPA, and Enricher. So these are probably three of the most commonly used tools here at the institution for just like generating pathway lists. And what we have found is that the differences are really dramatic. So this is just one example of one of the assessments we're doing. We took the gene list from 50 Go biological process entries. So these are annotated biological processes and pathways out of the Go biological process list. We picked them at random. The only requirement was we wanted them to be between, a reasonable size. We took things that were from several genes in size up to, I think, about 200 genes in size. We didn't go after things that were huge or things that were like literally 2 genes. But other than that, these were randomly selected. We ran them through these 3 tools, as I mentioned, and then humans went back and assessed whether the tool actually managed to get the pathway back or something like the pathway that was entered. And you can see how dramatically different the results were in this case. Enricher, and we did it both ways. It's kind of unfair to put in a pathway from Go Biological Processes to a tool where Go Biological Processes is one of the underlying knowledge bases being screened, but we did it both ways. We did it with and without Go. You can see that Enricher with Go got all 50 pathways back. Even without Go, using KEGG, Reactome, other pathway knowledge bases, it got 44. So very good. IPA only got 26 out of 50, and it didn't matter whether we turned on Go or not as part of the underlying knowledge base. So this had us a little confused. But the really striking result was GSEA. We basically ran this every permutation imaginable, and no matter what we did, unless we go to like a really, really ridiculously open nominal p-value, you get hardly any of the results back, even if Go is part of the underlying knowledge base. This was really, really striking. So it's not that the stuff that came back from GSA was wrong, it's that it was missing an enormous amount of information that the other tools were able to detect relatively easily. So this is the sort of thing, this is just one assessment. We're doing many different types of assessments. We're going to put these results up on the website, as I said, on the portal site at some point here in the not too distant future. And then, like I said, we'll probably publish the assessment methods themselves as well. So we're hopeful that this is the sort of thing that will help in groups making decisions in terms of what types of analytics that they want to use for some of these omics assessments. Okay, so the last thing I have here, checking my time. Not sure how we're doing. Anyways, okay, we're fine. So improvements to our infrastructure. This is another thing that was just really a necessity in merging GTAC and MGI. We had a lot of old legacy systems that were in place from the 2 groups. And frankly, trying to use either one of those moving forward just wasn't going to work. So we have embarked on and are now have some of these coming to fruition, a number of different interfaces, web pages, tools, software underneath, some of which you won't see necessarily directly, but will hopefully impact your experiences working with GTAC moving forward. So we have we put in place an appointment tool. So as you know, single cell requires an appointment because you have to bring viable cells. So this has to be scheduled in advance. So that someone's there to receive the cells and work with them immediately. So Jen's group has put an appointment tool in place. I'm pretty sure, Jen, correct me if I'm wrong at the end of this, but pretty sure that's already in use and that individuals can come in and make their appointments through that system. We also are going to be bringing online a new project and sample submission system for the rest of our services. And this will allow, it's basically a new ordering system for, like I said, for the vast majority of the services. The system's in testing now, and we're expecting to launch it in roughly the May timeframe. We're hopeful this will help alleviate a lot of the issues we've had in the past with project and sample submissions. And obviously, if there are issues, we will correct them and get them going. So basically get them working better as we go. But this is going to replace the old ordering and sample submission systems. Internally, we also have customer relations management software and new LIMS that we're working on. The purpose of these is to improve our communications with folks about their samples, where they're at, what part of processing, when they can expect results. We get asked all the time, where are my samples at? When can I expect the results, that sort of thing. So these systems will greatly enable improved communications and efficiencies for not only getting samples through the system, but our communications. And we expect the customer relations management software to launch in July. The LIMS software is work in progress. That's going to take some time. And I should have taken the LIMS off this. This first was really about the CRMS. The LIMS is work in progress. We have a number of different labs that have to have their components built. We are doing that now. I am hopeful that by the end of this calendar year, our new LIMS will be functional and in place for most of our laboratories. Again, we had to replace 2 different legacy LIMS systems from MGI and GTAC. And so, bringing all of the labs together into a single LIMS system has been a Herculean task, but we are making good progress, and we hope to have something functional before the end of this year. And as I had mentioned, we're also migrating GTAC to the new research computing environment. This has been a multi-year migration plan. We had 30 years of legacy data and workflows that had to be migrated, but it's well underway. We have added Dragon computers to an already generally faster compute environment. And so we're hopeful this will also reduce your bioinformatic processing time if you're having us do any bioinformatics processing for you. So turnaround times for alignments and gene expression and stuff like that should improve as we get everything moved over, because they should just run faster. It's that simple. So this will also shorten turnaround time to get people's data back. And the fact that a lot of labs work within the new research computing environment will hopefully make data transfers relatively quick and easy as well. So all of these things are really designed to improve efficiencies everywhere from our communications and interactions, submissions, all the way through the process of getting data back. Okay. So services and contacts. So today I've really only talked about new services. We have a substantial number of services that I didn't even touch on. Many of you are probably familiar with a lot of those services, but if you're not, you can go to our website, which is very simple, gtac.westall.edu. And on the front page, you scroll down a little bit, there's a laboratory services section, and that will take you to a detailed list of the different laboratories, as well as the lab manager. And you can, if you've got a question, you can either click on the laboratory itself to see detailed lists of services, or you can contact the laboratory manager if you have questions from within the site. So anything even beyond, the day-to-day stuff that we do or have been doing for a long time is all captured within the website. And so if you have any questions, you can probably find answers in there. And if you can't find an answer in there, you can probably find the contact of who you can talk to get you answers. Okay. So as Seth had mentioned, so that's pretty much the end of the GTAC discussion. Seth had mentioned this is going to be the first of a seminar series, and these are going to occur. The plan is for these to occur roughly monthly. You may see a broader announcement for this coming from Jeff Milbrandt. This one was one we already had planned before the idea for the full seminar series came online. So we decided to sort of just kind of roll it into that. But you may hear more about the seminar series from Jeff and the rest of the MGI moving forward. All of the different technology centers will likely give presentations, overview presentations, GESC, MTAC, which is the new mass spec center, 5, which is our functional genomics group, technology development, and basically any of the groups within the MGI. There will also be deep dive seminars on specific technology platforms. So, we talked about SomaScan today, but there's, we will likely have at some point a deep dive on SomaScan. There will be deep dives on probably 2 or 3 different GTAC technologies. Once we have the spatial up and running, we'll probably do a deep dive seminar on that. And I'm sure each of the other centers, technology centers, will also have some deep dive seminars as well. So that's all going to be forthcoming. And with that, I will stop. And if there are any questions in the parking lot, or if we want to go to verbal questions, that's fine too.
Thank you, Rich. One thing, Katrina Fronick, who oversees the Oxford Nanopore Laboratory, wants to make a quick comment concerning that technology. Katrina, are you there?
Yep, I'm here, Seth. We have a GridION in our technology development group. It hasn't quite been focused on long read technologies quite yet, but if there is interest in maybe lower throughput or checking out that technology, you can contact us and we can discuss your project.
Thanks, Katrina. Zihang had a question. You can talk if you like. All right, he raised his hand, but then he promptly went to get a drink. Jenny would like to know, is the February seminar detailing the bioinformatics collaboration available to view? I can't remember if we recorded that. Rich, do you know?
I don't remember. I have,
Okay,
and it was just and I didn't have any slides either because we just showed the portal. We went right into the portal.
right.
The portal's pretty self-explanatory, so we can definitely give you the link to the portal, and you can go in and kind of browse through it yourself. It's pretty straightforward, and there's information about there in there about how to actually submit a collaboration request or to look at the resources. If you're interested in becoming a contributor, there's a contributor registration up in the upper right corner. And basically there's some very simple information about how to register as a contributor. So, or if you just have questions and want to send me an email, that's fine too.
I believe on the same subject, Anna wanted to know, is this link to the RIS resources?
Which link?
Yeah, I'm assuming that she meant Anna. You're welcome to ask yourself if you'd like, but I believe she was talking about one of the links.
Sorry, it was actually misspelled. But I meant the research infrastructure services
Yes.
at WashU. So they do have a lot of compute platforms and help for computing. So I was wondering if your new platform or resource or portal is linked to them, or this is a completely different thing.
No, the resource portal is separate from RIS. This is something that, like I said, GTAC at MGI is maintaining the website, but it's meant as an institutional resource. It's a relatively simple website with a fairly simple database on the back end, and we're hosting it.
Okay.
That said, the research computing environment that we are migrating all of our main workflows into, that is the research computing environment that RIS oversees.
Okay.
So we're doing that with so all of these all of our main workflow migration stuff is being moved into that environment that they oversee.
Okay, and actually a follow-up question is your bioinformatics labs from Paul Clifton, is that something similar to what the RIS does also, like helping us? I was working with them to develop pipelines for RNA-seq analysis. So I was wondering if your bioinformatics section would do something like that too.
So we have workflows already developed for RNA-seq. And in fact, if I'm not mistaken, there might be a link to one of our resources on the portal. I could
not access the portal. The website that I have, or in your email, says not working for me.
Are you working off-site?
Oh, yes, I'm at home right now.
You have to VPN into the firewall in order to be able to access it.
Didn't catch that.
Yep. So if you VPN in, you should be able to get to the portal. There's a whole bunch of resources there, including some workflows and some pipelines. I'm not sure if we have as you can imagine, we have a very, very robust RNA-seq workflow. We process literally tens of thousands of RNA-seq samples a year. And I'm not sure if we have it in there. If we don't, we could probably make at least components of it available, at least what we use and how we use it.
Okay, thank you so much.
Rich, Tomas, who, by the way, I've unmuted you if you want to supplement this question, wanted to ask about your cross-pathway analysis comparison. How does the precision compare? Is there an increased prevalence of false positives?
So that is, so as I mentioned, the one figure that I showed, that's a sensitivity test. We have about a half a dozen other things we're doing, and some of the things that we're doing are to answer that question. So we have, for example, we're adding in, we're taking real world data and we're sort of opening the window and adding noise to see where, you know, you start picking up stuff that wasn't part of the original biology. And so that's actually one of the other analyses that we're adding into this overall umbrella. So like I said, there are about a half a dozen different things we're doing. And while the one I showed was specifically for sensitivity, we have another one that we're doing that very much looks at specificity.
Okay, fantastic. That is all the questions that came in. Tomas says, "Gotcha, thanks." So I think that's going to be the end of our talk. You can see in the chat box my email address is there. The website is there where you can put questions. We're always ready to field questions. People who are asking about what the list of the 7,000 SomaLogic analytes, please email me and I can send you that list. Yep. But other than that, I just can't think of anything else to talk about.
Well, I think we're just about at our time block, so that worked out great. 4 minutes to spare. So I just want to thank everyone for joining. I saw we had 120 folks on at one point, so even still 81 on now. So thank you everyone for joining the talk. And like I said, if you have other questions, please don't hesitate to email Seth or myself or Lucinda or Mike or Paul or whoever from the leadership group or the lab managers themselves.
All right.
Okay.